Hello students, here we are to complete the life history of Salpa. So this is the life history of Salpa 2, the second lecture. So let's begin. Each oozoid develops a complex tube-like stolon at the posterior end of its endostyle by a process of gemmation. Now this gemmation is a kind of budding, the formation of a new individual from some projection which grows out in the parent's body. So in the stolon also there are some cells which they develop into the new individuals. So this formation of new individuals by budding, kind of budding, is known as gemmation. This stolon actually consists of processes from all important organs of the body forming an endodermal tube. This tube, it has two peribranchial tubes, a neural tube, two blood sinuses, mesoblast cells, and a genital cord, all enclosed within an ectodermal investment. See, all these structures, they are helping in the formation of the new animal. Later, the stolon elongates, segments into a series of buds, penetrates through the parent test, and projects outside near the atrial aperture. The buds on the stolon undergo development, and those at its free end, being more advanced, detach in groups or chains. Each bud of such a chain ultimately develops into a salpa of the gregaria or sexual phase. Now, if you do not remember, let me tell you, the stolon, it was the extension of the ventral posterior side of the endostyle, and this posterior side of the endostyle, it forms the stolon, and as explained above, the stolon, it is a tube-like structure which is having all these structures, these two peribranchial tubes, neural tube, two blood sinuses, mesoblast cells, and a genital cord, and they are all enclosed within an ectodermal investment. So, and these all structures, they combine, they are with the combined effort, the cells are formed which differentiate into new individuals. And the tip of the stolon, it is having the most matured individual, and it can stay with the chain, or it can be separated from the chain to live a free individual life. Now, the stolon which gives rise to new individuals, the stolon is a part of the asexual phase of the salpa. And now when it's by budding, when it's giving rise to new individuals, these individuals are the gregaria phase of salpa. Means these individuals, they are not having a stolon, they are having the sexual organs which interact to form the new individuals. So it is a sexual phase of salpa. So the salpa of the gregaria phase may remain attached to the chain or may get detached from it. Chain means the stolon. Its cylindrical body is ovoid or fusiform, as you can see, and smaller than the salpa of solitaria phase. It is, you can see, it is smaller than the solitaria phase. It is invariably asymmetrical and have fewer muscle bands. These are the muscle bands 1, 2, 3, 4, and definitely they are less than what they were in the solitary phase. It never develops a stolon but contains gonads. A testes lying ventrally. Here you can see this is the ovary, and there is no mention of the testes, but it is lying here in the loop of the intestine, lying ventrally in the intestinal loop, and an ovary on the right side. This here, here somewhere here is the intestinal loop, and the ovary is present on its right side. The gonads are protogynous, that is, the ova usually only one at a time mature first. The development of fertilized ovum takes place inside the parent body within a diverticulum. This, when I was doing the diagram, the sagittal section from the left side, if you remember, there were two cavities overlapping in the center, the branchial and the atrial cavity, and there was testes was lying near the stomach in the intestinal loop, but the ovary, it was present in the diverticulum in the atrial sac. You can go back and see the diagram again, and you will understand this point. The development of fertilized ovum takes place inside the parent body within a diverticulum uterine sac of the atrial cavity. The development is direct. There is no larval stage, and the segmentation is holoblastic. In the last animal, Pyrosoma, we had seen that the segmentation is meroblastic, and I will explain to you what is meroblastic. And now holoblastic, you must be knowing holoblastic. Holo means full, blastic means division. So the division takes place in the whole of the egg, as compared to Pyrosoma, where it was only in the blastodisc due to presence of a large amount of yolk. At an early stage, the embryo develops a central placenta through which it derives nourishment from the body of the parent. Somewhat later, some special cells called elioblast cells, you can see them here in the diagram, the elioblast cells, which are probably remnants of vestigial tail, develop at the posterior end of the embryo, and become filled with oil globules, which serve as reserve food for use in later development. Ultimately, this embryo develops into a asexual solitaria salpa. Here you can see this is an individual which is formed from the stolon. You can see the individual. There is the branchial aperture. This is the branchial aperture, and this is the posterior side, and this is the branchial cavity. This is the atrial cavity, and here you can see the dorsal lamina, the dorsal tubercle, nerve ganglion, and this is the atrial aperture. Here near the atrial cavity, this is the atrial aperture, and on its posterior side, this is a new individual forming by the help of the placenta, and later on the help of these elioblast cells. So slowly this will convert into a new individual. Here you can see the placenta, and this is the stolon. Ultimately, this embryo develops into an asexual solitaria salpa, which leaves the body of the parent. This is the body of the parent. So when the individual is formed, it leaves the body of the parent and because a young one is being born from the individual, there is no laying of egg or something like that, so this is considered to be a viviparous animal, and after it is free from the parent, it swims about and gives rise to a ventral stolon, and thus commences to form individuals of the sexual gregaria phase. When it gives rise to a ventral stolon, it becomes the solitaria phase animal, and then this solitaria phase of animal, it starts preparing itself to give rise to the individuals of the sexual gregaria phase, and this way the life cycle. So you can see in this diagram the asexual solitaria phase. With the help of the ventral stolon, in the ventral stolon the buds are formed, and these buds they separate to form individuals which are of the gregaria phase, and this is the sexual phase, which is having the gonads. The gonads are fertilized ovum and testes and the ovum, and the fertilized ovum, it gives rise to the embryo, and this embryo again forms the solitaria phase. Thus the sexual solitaria phase and sexual gregaria phase, both differing in structure and mode of origin, alternate regularly to Salpa's life cycle as illustrated in the diagram. So this completes the animal Salpa, and with this I complete the third topic of unit 1 of paper 11, and now we'll move on to the next topic. So thank you, and be prepared to do the next topic, which is the origin and evolution of vertebrates. So thank you once again.